Hello, thanks to the organizers for inviting me to tell you about how Pfizer Worldwide Research has been using 7Bridges data science investments to support our hybrid embedded bioinformatic work. And I'm mindful that I'm the last to talk before you either break for dinner or for lunch. And so I hope this is enjoyable and an interesting perspective about the infrastructure requirements that we all need to do modern clinical omics. So at Pfizer, Worldwide Research and Development's bioinformatic needs are split across 8 different core topic-focused practitioner groups. And this is pretty common to large organizations. So like many others, be them companies, institutes, or collaborations, this different area of focus for our bioinformatic and clinical omics scientists can really determine different computational infrastructure needs. And these different needs present unique challenges for professionally managing all of the diverse projects that are being done by all of the wonderful researchers in many different areas of expertise like we've seen today. This model is known as a common embedded model for data science or bioinformatics. And because of it, we face common problems or headaches with it. The needs stemming from embedded teams are diverse, but each of the teams face common headaches. However, because the teams are based in different locations, working on different topics, these common headaches can sometimes be solved using different approaches. This is a core challenge of embedded data science and bioinformatic groups. The deep collaboration that embedding groups supports with diverse non-bioinformatics scientists is wonderful, but it risks siloization between bioinformatic and clinical omic practitioners. And so to address this challenge at Pfizer, the groups have self-arranged into a core matrix organization that we call integrative biology. Integrative biology acts as a unified central group to solve the community needs of all bioinformaticians. By organizing ourselves into this hybrid model, we've actually been able to embark on working to meeting many of our core needs, having modern computational infrastructure that supports clinical omics in the 2020s, as it really is today. So this work is really stemming from an effort to fight the siloization that comes from this model. And in 2019, we actually formed a formal project to work on this, to develop clinical omics solutions for all of our core needs. Primarily, we found that the modern omics we do today were highly collaborative, highly integrative, and highly dynamic. And most especially, they were project driven by the scientists working together to meet a common scientific question, not workflow driven. And that really highlighted the diverse needs that we all had across worldwide research and development. In particular, as the team worked to define our needs, they focused on 4 domains: computational environment, data visualization, workflows, and data science. And it very quickly became clear that in the 4 needs, 3 of them were highly specifically integrated. So the computational and data storage environments needed to be flexible and scalable enough to support our rapidly evolving workflows. We can't disentangle these needs. They're really coming together, and the solution that also comes together. One of the primary case studies that drove this need of an integrated environment that supports storage and computation was from early adopters of single-cell RNA-seq, who had all begun to develop local solutions. So several years before this effort had started, they had each established local, bespoke, but diverse solutions to address their rapidly expanding data generation. And that's because the type of data that single-cell RNA-seq uses requires an order of magnitude more sequencing than previous methods. Yet each of these local bespoke models that early adopters had developed was already running out of storage and computational capacity. it was already failing. And so it was clear a sustainable solution for all was really needed. In defining the needs, though, it was clear that the gaps were quite enormous. For data storage, they were focused around a lack of centralization, a lack of API access of the data, and little ease of sharing data between different groups. There was also a massive scalability of data because it was clear that groups were generating data at a much higher pace than they had been doing in past years. And the recommendation, generally speaking, was to consider pivoting to off-premises solutions, meaning cloud-based storage solutions that could accommodate and naturally support this type of modern genomics work. Similarly, for the computational environments, assessments of our needs and goals was very analogous. The needs were, again, resources and collaboration. The objective was to facilitate that collaboration, and the recommendation was to find a cloud partner who could build these systems. And one thing that really came up as we were discussing the need of a cloud partner was what that solution would look like. So the optimal solution would really need to balance many considerations, and it would need to be user-friendly for scientists who are topic-focused, not IT or infrastructure-focused. Any cloud-based solution should reflect that modern flexibility of data science needs is going to surface again and again, and we needed sustainable and customizable solutions that could be co-developed rapidly. And we also needed the solution to foster data science best practices and maintain data privacy standards with the highest amount of data security. So collectively, teams took on the opportunity to build this out and find someone who could fulfill these needs for us. Specifically, in 2019, a large team listed here assembled to take the task on of solving these needs. They kicked off an RFP that resulted in partnership with 7 Bridges, a cloud-based platform for bioinformatics data science, genomics, and clinical omics. And this work with 7 Bridges has become a foundation for our vision of modern clinical infrastructure. Now, what did they do? Well, over the course of a short period, that team worked with 7 Bridges to deliver a multi-cloud, project-centric, flexible storage and computational platform. And they did it in under a year, which is pretty remarkable given the scale of need that we were facing. Now, what does the solution with 7 Bridges look like, and what did we actually do? Well, it looks like a project gallery that centralizes our access to datasets and projects, and is fundamentally user-friendly for all the practitioners across worldwide research and development. This platform is built on a core 7 Bridges project concept. It uses all of the features of 7 Bridges, and it fundamentally made sharing and collaboration of work possible by representing all of Pfizer work in bioinformatics and clinical omics in a single project gallery landing page. This project gallery then supported basic but consistent workflows like searching for projects with the same topic or name or creator. Underneath it, though, we had access to both AWS and Google Cloud storage and compute that were all owned and controlled by Pfizer so that it met all of our privacy and data security needs. It also enabled API access to all of the data and code to support ongoing enterprise-level data science and machine learning efforts. Finally, it really enabled sharing with access requirements for all of our scientists. So every project listed here in the project gallery has an option to request access to it. And that means that all scientists can request to participate in a project that they're collaborating on. But if they're not participants, it's still visible to them. And fundamentally at Pfizer, that represented a shift from a default unshared, unknown project gallery to a default visible project gallery. If you're a bioinformatician at Pfizer, you can see all of the projects that other Pfizer bioinformaticians are doing without getting access to the access restricted data itself. Additionally, we were quite excited to be able to have this based in working on GCP and AWS systems, which can be a challenge to navigate if you have data coming from different sources or stored on different systems. It's also important to note that all projects in this gallery are tagged for discoverability themselves. So the project gallery serves to organize all of our work through metadata via different tags that bioinformaticians put together. The tags are added to projects and reflect high-level metadata like disease, tissue, strain, etc. And these tags enable searchability of projects across integrative biology. And they also enable sharing of projects to outside groups. So that means that non-Pfizer scientists can join and run work in this environment with no pain points. The system is the same for them as it is for Pfizer, but the security standards are fully maintained. And so we've had this project gallery up and running across WRDM, Worldwide Research and Development, for 6 months now. Its impact has been very clear rapidly. By having a centralized storage and computational access point, we've removed numerous past problems, like needing to move data from one cloud partner to another, or having code break between time or different groups or different environments. The lack of these pain points has accelerated our projects. It's enabled improved collaboration within Pfizer and outside of Pfizer, and it's supported ongoing additional investments that actually build on top of this project gallery concept. Feedback from scientists really suggests that this is a huge improvement over past solutions, and particularly that Seven Bridges offers all of the flexibility of group-owned cloud platform accounts without the burden of self-maintenance. And this is particularly valuable to us because we have bioinformaticians who are science-facing, who are working on science with other discovery and development scientists, and who don't have the IT expertise to maintain virtual machine or cloud infrastructure accounts themselves directly, but who don't want to be slowed down by needing to work with infrastructure experts to really modify those environments in a day-to-day way. And so for us, this has represented a massive improvement in what we see as our needs as computational scientists across the organization. And we think that this platform could solve similar challenges for other diverse groups who work in clinical omics across the practitioners that they have already. And so with that, I'll accept any questions that we might have. I'm wrapping up a bit early, but hopefully that time is appreciated and returned for others. I'm happy to take any questions at this time. Pippa, I'm not seeing any in the chat. All right. Well, thanks so much to the organizers for inviting me to speak today. I very much appreciate it. And I hope that this tells you a little bit about how Pfizer is approaching our infrastructure to support all of our proprietary bold moves in computational science. Thanks so much.